Dear students, welcome to Top Talks on Plants. In this very lecture, we will introduce the hybridization. Then we will have an idea about the types of hybridization. Then we will have a detailed discussion on the process and techniques through which we can achieve hybridization. But before that, we should know the how we should why should we know about hybridization. Dear students, you know that the world population is closely related to with the agriculture sector, and where the agriculture sector acts as the major source of food. And the current global population is 7.6 billion, and it is expected to be 9.2 billion in 2050. And by 2050, the population in the developing countries will be roughly 8 billion, and the population in the developed countries would be 1.2 billion. And notably, this burgeoning world population would require 70% more food. And it is very interesting to note that agriculture remains central to the world economy, and 60% of the population depends on the agriculture for survival. However, the general consensus is that global agriculture production has to be increased by about 60 to 70% from the current levels to meet the increased food demand in 2050. So, to meet this increasing demand of food, agriculture sector is under great pressure to increasing the crop productivity. So, in order to do that, though we have the and are working on increasing high yielding crop varieties and also exploring crop resistant to major biotic abiotic stresses. However, the mostly a range of chemicals are being used at various levels in agriculture in order to improve both health and productivity of crop plants. And agriculture, the chemicals are defined as the chemicals such as pesticides, herbicides, fungicides, insecticides, and fertilizers are used in agriculture to control pests and diseases or control and promote growth. And these chemicals used in turn are impacting the health of the soil as well as the plant. So, how we would feed the world population? What does it mean? It means that in 2050 the arable land will increase by about 200 billion million hectares, mainly from sub-Saharan Africa and Latin America. And this expansion may come at a heavy price as deforestation in the Amazon, for example, and would cause severe ecological upheavals and release of more carbon stored in the soils to the atmosphere. So then, what should we do? There, we have to explore approaches in order to improve the crop health. So there are a number of methods for crop improvement, and the methods are the plant introduction and the crop, plant breeding, acclimatization, tissue culture, genetic engineering, and polyploidy. Today, we will discuss in detail the crop or plant breeding approach, that is the hybridization. So, crop or plant breeding is nothing but it is the science of changing the traits of plants in order to produce desired characteristics used to improve the quality of nutrition in products for humans and animals, and also aims to produce crop varieties that boast unique and superior traits for a variety of agricultural applications, and through which we can achieve improved or high plant crop yield. On the other hand, the plant breeding or crop breeding can be accomplished through many different techniques. Simply selecting plants with desirable characteristics for the propagation, that we can say the selection method, and two methods that make use of knowledge of genetics and chromosomes, and two more complex molecular techniques. So let's have a detailed idea about the hybridization. So hybridization is one of the stages in plant breeding process, and it is the mating or crossing of two plants or lines of dissimilar genotype, and hybridization mainly aims to develop new genotypes evaluate their performance, exploit hybrid vigor, and hybridization helps in combining characters of different plants. And I wish to mention here that through hybridization, no change in genetic content of organism occurs. Instead, hybridization produces new combinations of genes. Before going into any detail, I want you people to know about a brief history of hybridization. That means the first natural hybridization that occurred during the year 1716, and it was reported in corn by Cotton Mather. He was a New England Puritan minister, prolific author, and pamphleteer, and is remembered today chiefly for his book Magnalia Christi Americana that was published in 1782, and he contributed to plant hybridization and also to the promotion of inoculation as a means of preventing smallpox and other infectious diseases. And the first artificial interspecific hybrid was produced by Thomas Fairchild in the year 1717. And Thomas Fairchild was an English gardener, and he was the first person to scientifically produce an artificial hybrid, that is Dianthus caryophyllus barbatus, in the year 1717, and it is popularly known as the Fairchild's Mule, and it is a cross between a Sweet William and Carnation Pink. And the Thomas Fairchild corresponded with Carl Linnaeus and helped by experiments to establish the existence of sex in plants. So the Dianthus caryophyllus barbatus belongs to the family Caryophyllaceae, and it is native mainly to Europe and Asia. The third point under the heading of brief historical aspect, I want to mention here that the practical utilization of hybridization that occurred in the year 1760, and it was reported by Joseph Gottlieb Kohlreuter. He was a German botanist and pioneer in the study of plant hybrids, and he was the first to develop a scientific application of the discovery made in 1694 by the German botanist Rudolf Jacob of the sex in plants. So, regarding the types of hybridization, based on the taxonomic relationships of two parents, we can categorize the hybridization into two major types here. That is the intervarietal hybridization and the distance hybridization. What does it mean? In the intervarietal hybridization, the parents involved in hybridization belong to the same species. They may be two strains, varieties, or rare races of same species. So it is also known as intraspecific hybridization. So an example would be crossing of two varieties of wheat, that is Triticum aestivum, rice, that is Oryza sativa, and some other crops. So intervarietal crosses may be simple or complex depending upon the number of parents involved. And in the distance hybridization includes crosses between different species of the same genus or of different genera. And when two species of the same genus are crossed, it is known as interspecific hybridization. But when they belong to two different genera, it is known as the intergeneric hybridization. And generally, the objective of such crosses is to transfer one or few simply inherited characters like disease resistance or drought tolerance in the crop species. And sometimes interspecific hybridization may be used for developing new variety, such as Clinton oat variety was developed from a cross between Avena sativa and Avena byzantia, both haploid oat species. And we can cite another example, that is the triticosicale, or it is a hybrid of the wheat and rye, that is wheat triticum and rye sickle. Another example can be cited here, that is a radish and the cabbage, that makes actually the rabbage, Raphano brassica. So intervarietal hybridization can be simple or simple cross or the complex cross. A simple cross means two parents are crossed to produce the F1, and F1 is self to produce F2 or used in a backcross program. On the other hand, the complex cross uh, involves the more than two parents, and here the two parents are crossed to produce the hybrid, which is then used to produce F2, or they are used in a backcross. So regarding the techniques through which we can achieve the hybridization, it involves 8 major steps. That is selection of parents, selfing of parents, emasculation, bagging, tagging, crossing, harvesting and storage of the F1 seeds, and the last one is the raising the F1 generation. So regarding the selection of parents here, selection of parents, that is the desirable male and female parents are selected here, and parents should be healthy and vigorous, and the selection of parents depends on the aims and objective of the breeding program, and we should keep in mind that both parents mature at the same time, and parental plants must be selected from the local areas and are supposed to be the best suited to the existing conditions. The second the approach is the selfing of parents or artificial self pollination. So it is required for inducing homozygosity of for eliminating the undesirable characters and obtaining inbreeds, and also required for the for inbreeds. An artificial self pollination is done in the bisexual flowers covered with the paper bags and before anthesis in order to avoid natural cross pollination, and self pollination crop occurs inside the bag, where in the cross pollinated crops the male and female flowers are bagged separately before anthesis, and when stigmas become receptive, pollen grains are collected and brushed on stigma, and in this way we achieve the artificial pollination. The third approach is the emasculation. What does it mean, emasculation? The inbreeds are grown under normal conditions and are emasculated. So emasculation is the removal of stamens from female parents before they burst and shed their pollens. That means the removal of the male part here. And emasculation can be defined as the removal of stamens or anthers or the killing of the pollen grains of a flower without affecting in any way the female reproductive organ. And emasculation is not required in unisexual plants, but it is essential in bisexual or self-pollinated plants. And we have several approaches to achieve the emasculation. That means we can obtain emasculation through hand emasculation or forceps, scissor method, or hot water treatment, or cold water treatment, alcohol treatment, suction method, and through which naturally through the male sterility or cell incompatibility. And we can also use the chemical gameticides that we will have idea about in the next slides. So in the hand emasculation or forceps or scissor method, generally this method is generally used in those plants which have large flowers, and corolla of the selected flowers is opened and the anthers carefully removed with the help of fine tip forceps. In the second method for obtaining the emasculation, we can go for the hot water treatment. That means it is done in minute flowers, and here in such hermaphrodite flowers, for example in the bachara jowar, emasculation is done by dipping the flowers in hot water for a certain duration, for example 1 to 10 minutes of time, and actually time varies from species, and we have to standardize this approach in particular plant. And hot water treatment actually the method is based on the fact that Gynoecea can withstand the hot temperature at which the anthers are killed. Another approach can be used here of the cold water treatment in order to achieve the emasculation. And cold water also kills pollen grains without damaging the gynecium. And in rice, actually, 0 to 6 degrees Celsius temperature is maintained to kill the pollen grains. And however, cold water treatment is less effective than the hot water treatment in obtaining the emasculation. Another approach we can adopt to obtain the emasculation, to achieve the emasculation, is the alcohol treatment method. Here, what happens here? The flowers or inflorescence are immersed in alcohol of a suitable concentration for a brief period. For example, in alfalfa, a treatment of even 10 seconds with 57% alcohol is sufficient to kill the pollen grains. However, alcohol treatment method is not commonly used for emasculation because duration of treatment is an important factor, since a very short duration is required, failing which even the gynecium may be damaged. Another approach through which we can achieve the emasculation is the suction method. Here what happens in the suction method, it is actually the mechanical method and is suitable for the crop having minute flowers, and the amount of pressure is applied in such a way that only anthers are sucked out and other parts of the flower like gynecium remain intact. However, here 10 to 15% of self-pollination takes place in this process. Another approach is the through the male sterility or self-incumbability method. So here emasculation option can be eliminated by the use of male sterile plants. And in some self-pollinated plants, or for example sorghum, onion, barley, anthers are sterile and do not produce any viable pollens. Similarly, in self-compatibility may also be used to avoid emasculation. The last approach is the chemical gameticides. That means we can use certain chemicals, those are capable of causing male sterility when sprayed before flowering. We can cite here the chemical gameticides as 2,4-D and naphthalene acetic acid, malic hydrazide, and tribenzoic acid. The fourth approach in achieving the hybridization is the bagging after the emasculation. The fourth step and emasculated flowers or inflorescence is immediately bagged to avoid pollination by any foreign pollen, and the bags may be made of paper, butter paper, glycine, or fine cloth, and butter paper or vegetable parchment bags are most commonly used. So here the bags are tied to the base of the inflorescence or to the stalk of the flower as shown in the picture here. On the left hand side here, the bagging is done with the emasculation in bisexual plants and before the stigma receptivity and dehiscence of the anthers in unisexual plants. And both male and female flowers are bagged separately to prevent contamination in male flowers and cross-pollination in female flowers. Another step is the tagging, the fifth one step, the tagging. Here the emasculated flowers and bagged flowers are tagged just after bagging, and generally circular tags of about 3 centimeter or rectangular tags of about 3 into 2 centimeter are used, and the tags are attached to the base of the flower for the inflorescence with the help of the thread. Here the major things that we should mention on the tag includes here the number of referring to the field record, date of emasculation, date of crossing, and name of the female parent is written first followed by a cross sign, and then the male parent we have to mention there. For example, C and D denotes C into D denotes that C is the female parent and D is the male parent. Here we have to mention these information on the tags. Another step in achieving the hybridization is the crossing. So it is the artificial cross pollination between the genetically unlike plants, and herein mature, fertile, and viable pollens from the male parents are placed on the receptive stigma of emasculated flowers to bring about fertilization, and pollen grains are collected in petri dishes, for example in the case of wheat and cotton, or in the paper bags, for example from the maize, and applied to the receptive stigmas with the help of a camel hairbrush, piece of paper, toothpick, or forceps. And in some crops, for example, jowar and bajra, the inflorescence of both parents are enclosed in the same bag, and after crossing, the flowers are bagged again. And the seventh step in the achieving the hybridization is the harvesting and storing the F1 seeds. That means the cross heads or parts of desirable plants are harvested, and after complete drying they are threshed, and seeds are stored properly with original tags. And the last one, the last step is the raising the F1 generation. That means in the coming season the stored seeds are sown separately to raise the F1 generation, and the plants of F1 generation are progenies of cross seeds and therefore are called the hybrids here. So in this way we have achieved the process of hybridization. Okay, so thank you very much.